attempts at extraction of information. Presently it's 7 pages. I expect it will be about 8.
When you ask questions about the papers, are you going to be going from generally just one paper, or are you going to be going from multiple?
Presently, there is one multi-part question from one paper. There will be at least that. There may be another. I'm having a hard time thinking of questions from the other paper. Those will expect something like last test. Is everyone simply resigned to their fate?
Can you let us know which page?
I could.
We're just will you?
So when you mean distinguishing features,
Yeah, in the question, it does tell you what paper it comes from. Okay.
then for distinguishing
At present.
between viruses, are you just going to do like double-stranded versus single-stranded DNA or RNA, or like what do you mean by features?
So at the start of every chapter, there's a description of the virus that I've encapsulated by saying these are minus-strand RNA viruses. So single-strand, double-strand type of nucleic acid, whether it's the sense or the antisense strand. Geometry, whether or not.
So everything in the blue box in the first.
Blue boxes are great
The
places to
blue
start.
boxes.
You know what else is a good place to go to?
The book.
Well, you could just memorize the text if you have eidentic memory. That would be my one suggestion. For those of us lacking, there's also a little encapsulation at the end of what you should know coming out of this.
Yeah.
When I'm thinking like, I need one more question, you know, okay, I'll ask about this. So there's highlights to hit there. I mean, this is general study tips you should have picked up when you were a freshman in high school. But just so you know, I am looking at those.
Smells like success.
Let's do this.
So, can you give us an example of like what kind of minutia we may be responsible for? Like, I'm just wondering to what.
Yeah, how minute?
Yeah.
Yes, I certainly can. What is the receptor for virus X? I'm not going to ask you what the gene names are for things. I might expect you to know what a gene name is being shown it. So,
Being shown.
and any gene name I can't come up with off the top of my head, I'm not going to ask you for. So, for example,
So all.
trying to find one that's got an obscure name. So, trying to look at. So GAG-Paul would be an example of a general retroviral gene that I would expect you to know. I'm trying to find a specific one. PB1, for example. I'm not going to, actually, polymerase basic one, but I wouldn't ask you to know a gene name from a specific class of virus, but you should know what TMSU is, for example. Is that helpful?
Yes.
Somewhat, perhaps.
And you said the breakdown is about the same as last time, so about that many matching multiple
Yes.
short answers?
Yes.
Would you have to draw anything?
Yes. You always have to draw something on the McMurray test.
Or would you have to draw a specific viron or a pathway?
Yeah, I think I'm struggling with thinking about like where the short answers are going to come from for this test, aside from one.
Yeah, I am too, but I've come up with a couple of doozies.
For example,
This is the kind of frustrating I'd like but hate to. I know what you want to know, and I got to not tell you what you want to know, but I want to tell you something helpful so you can figure out what so let's know something about the replication cycle, know how they differentiate from one another. So draw the general replicate. Don't write this down like it's verbatim because it may or may not be on there. So draw the replication cycle for influenza virus showing the following individual steps. From attachment tag. Or how does the rhabdovirus replication cycle differ from herpes virus? List 3 ways.
This is really hard, yeah. It's a lot of stuff.
Should we know how to draw like the different figures like last time?
Yeah, we can have to draw a graph.
Maybe. What graph would I ask?
The AIDS graph.
No
that we looked.
graph, no graph.
There's only one graph. And there are like 9 chapters.
That's not true.
The main graph.
I'm sure he'll pull something out of this.
So that I think, I mean, I'm having a hard time coming up with
Great, so I'm gonna fail.
draw a graph. What do you mean graph? Other than
Like you're offending me with that
this
bullshit.
is a graph.
That one.
Like something similar to the,
Which one?
like something similar to the question we had for, post-grad, the binding of.
Oh, sure. I can think of something like that. I'd give you that.
I still need to do that graph. So what are you doing after this? I have a football game today.
Sorry, I don't know what. Oh, it's good. I'll try to think of a graph question.
I have to.
I can't think of one offhand.
When we talked about influenza, that's the only one we talked about.
Like you do CD4 binding.
I feel like the only real graph.
That one.
Yeah, that's the only graph I can think of that's in the text.
Oh, yes.
And-
I'm all over that one.
I could show you graphs that say interpret. Actually, I do that on the test. You have to interpret graph. Just thinking offhand, you know, I can't think of something offhand on this graph. Not to say I won't, but you know, what would this curve look like in an immunocompromised individual?
You mean somebody didn't have their immune system before they got HIV?
Yeah.
Yeah.
Let's get real creative.
I mean, that's an easy one, right? I couldn't think of a graph in the text beside this off the top of my head, and I couldn't think what to ask you. The point, when you see a graph, the graph conveys information. Like the point here is the balance between the immune response and the destruction of T cells. So if you had, I don't know, some sort of leukemia offhand, you wouldn't see, you would see, you'd have to explain. That's too easy, so I won't ask that.
So minutia, like one of them would be like we're not even gonna like.
Offhand, I would say you have a sufficient number of index cards.
But are they right?
Do we have to know the ways in which viruses invade the immune system?
I'm sorry?
Do we have to know the ways in which viruses invade?
That would be a great question.
Can you repeat that question?
So, list 3 mechanisms by which, do you want to do a particular virus? I don't know. Influenza might evade post-immune response. Design your own virus and its ways to evade. I don't know. It would certainly be a fair question.
I like that question.
Oh, Carlos, if you like it, I'll have to. Anything else?
Only like easy questions.
I forget where my markers are today, please remind me to put it like that. Okay, so just to reiterate, so feel free to completely tune me out now. None of this will be on the test, so from this point on, this is final exam stuff. And so this section of the course will be more heavily weighted on the exam than will stuff prior to this. But the exam will be comprehensive, so you're not allowed to forget anything. Keep your index cards. So bacterial viruses are known as bacteriophage, which comes from the Greek to eat, or phagus. So I guess you could say these are bacteria eaters. They are the sources of, well, in addition to being very important in the biology of bacteria, they're very important sources for us as molecular biologists and biochemists. They are the reason why we have a number of tools. So undoubtedly, since you've had genetics, you've used restriction enzymes. So bacteria have developed host responses to infection with phages, and a number of our toolkits today in the lab are a result of this interplay between bacterial viruses and host viruses. For example, restriction enzymes, EcoR1, BAMH1, etc., are enzymes produced by the bacteria to hydrolyze viral DNA before they kill the virus. And then there are a host of modification enzymes, for example, methylases, that we also use in the lab to do all sorts of molecular biology. Bacteria methylate their own DNA so that they don't get cleaved by restriction enzymes. So in addition to the basic biology, we have some research, commercial and industrial interest in viruses. The guest speaker, in fact, that I have booked for next week, although we're not totally confirmed yet, works for a local industry that is producing a bacteriophage product to put in yogurt, among other things, because they're also long been sought targets of therapies for bacterial infections. So the idea being, if you consume phage that are against, say, salmonella that were on your chicken that you ate last night, prior to gastroenteritis breaking out, a phage might kill the salmonella. So they're interesting from basic biology, laboratory research biology, and potential therapeutics, although like many therapeutics, they've let us down. Although perhaps Cobb County will be the point of origin of the next big phage product. They come in two flavors, virulent and temperate. And essentially the division is virulent always cause lysis or breakage of the cell. So you'll see in an animation later. Lytic phages, the adjectival form of lytic, cause the cell to burst open. Temperate phages can either cause lysis or lysogeny, which is the bacterial version of integration into the chromosome, like we see in HIV with the provirus integrating in the chromosome. Lysogeny is the process by which the phage genome inserts itself into the bacterial genome, which microbiologists like myself will call chromosome, but are not chromosomes. And then from that lysogenic state, they can either return to a lytic state at a later date, or persist. This is the worst classroom because the screen covers up half the board. jealous of the person writing on the other side of the board right now because I can hear her. She's got such a lot of whiteboard space. Okay, so there are several different flavors of phage. Let's start with the single-stranded RNA phage, of which MS2 is our example. They are small icosahedral. They are the plus strand of RNA. Their genomes are very small. The viruses themselves are small, 180 coat protein monomers. That's individual molecules, not types of proteins, and one maturation protein, one coat, which is known as A. So here you see just your standard spherical macromolecular but icosahedral shape virus, single-stranded RNA genome encapsulated. Being small, taking MS2 for example, they don't have a lot of information. There are 4 open reading frames with 4 genes. You see in the case of, this is just another one, another virus, but we have 4 open reading frames. One is in a different frame of reference than the coat replicase, so depending upon transcription, you either get coat, lysis protein. Replicase is the polymerase and maturase is that enzyme that's also incorporated in the capsid. They infect their cells via the pilus, which is a common theme. Bacteria engage in sex, as I'm sure you know, engineers entertaining other types. They can exchange nucleic acids through a structure called the pilus. The viral proteins, proteins attach via the pilus and enter, as we'll see. Regulation of expression. Like with other viruses, they'll need more of the structural proteins than they will of the enzymes. So regulation of expression is via secondary structure in the RNA. So let's take a step backward for a minute and realize that the host cells are prokaryotic. So we're not going to use any terms like nuclear transport. If there's no nucleus, there's no splicing because bacteria don't splice. Getting into the cell beyond the plasma or the cytoplasmic membrane of a bacterium gets it access to DNA replication, transcription, and translation machinery. So we have our plus-stranded RNA molecule, that genome length molecule is the messenger RNA. So immediately upon infection, you can have both translation and replication. Sorry. So it's going to need a lot of copies of coat protein relative to both maturase and replicase. Within the secondary structure, so when we look at this, what this is attempting to show you is that there's secondary structure. That means base pairing between strands on, so base pairing between bases on the same strand because there's only one strand. So for example, the A site, so this is the promoter for the maturase gene, is inaccessible, sorry, the ribosomal entry site is inaccessible to the ribosome because of this base parent. The coat protein site, the ribosomal binding site, is accessible such that translation can occur for coat protein prior to maturase. That coat protein is accessible, replicase, the enzyme that's going to replicate the genome is inaccessible. So the first thing that's going to be translated on upon infection is CP or coat protein translated. Thinking mechanistically, of course, the ribosome is binding to the ribosomal entry site, and then translation occurs in the 5' to 3' direction. If you follow through, you'll see that this is 5' to 3'. Downstream of the ribosomal entry site is the Min-Ju site. Sounds like the special at the French restaurant. The Min-Ju. It is what's known as a long-distance interaction. Or LDI. So the replicase site is inaccessible because of Min-Ju interactions. As the ribosome translates the coat protein gene, it's going to disrupt those Min-Ju base pairing interactions, making the replicase site accessible. So, regulation of replicase translation is translationally coupled to coat protein translation. So every time a coat protein is made, the replicase ribosomal binding site becomes accessible. That says translational coupling, sorry. No, we can't. It doesn't mean replicase necessarily is translated in that moment, but the more coat protein that gets made, the more replicase, the more potential for replicase translation to occur. A very small amount of lysis protein compared to coat protein is necessary. So this lysis protein is produced when there's slippage in the coat protein. Accumulation of coat protein represses replicase. Translation. Coat protein being translated turns on replicase. Coat protein having been translated in large quantities represses replicase by binding the RNA. So there's a push-pull, as it were, regulation of how much replicase is made, and there's a lot less replicase needed than coat protein. Replicase having been translated replicates the genome, which inhibits translation. That should make sense, because both always happen in the 5' to 3' direction. for a moment about what that means. So with our ribosomes are going this way, so if it's here, it's translating this way, opening up, either allowing for A or replicase translation. Replication of the genome is going to go 5' 3' relative to the daughter RNA. So we've got to make the minor strand of RNA. So replication is going to be going this direction while translation is going in the opposite direction. They would, in fact, inhibit one another. The story is a little more complicated than
Does one kick the other one off if they're both happening?
that. Effectively, yes. I don't know the details, but the replicase, so that the viral polymerase binds a ribosome. So not only is it, so the translation is going this direction, replication is going that direction. They're both 5', 3'. Although it's a little bit tricky because you're talking about the plus strand in one sense and the minus strand in the other sense. But that replicase is going to bind a ribosomal protein, which is going to inhibit formation of the large and small subunit in the translational complex. So as it's active, we're inhibiting translation via sequestering of the ribosome. So multiple replication daughter molecules can be made as minus RNA. So this, the plus strand that was the infectious molecule can either be template or package, or well, it has to be template. It can also go through replication several times. So we can have plus strand package, but if we go minus strand, replicase has to go the other direction and make the plus strand progeny molecules. So these are known as replicative intermediates. Or RIs. Replicative intermediate 1 is the plus strand. Is the minus strand. So that's relative to the template. Template here is the plus strand. This is replicative intermediate 1. The template here is the minus strand we're making plus. That's replicative intermediate 2. So during plus strand synthesis, this A site, so I'm sorry, let me back up. Because of during infection, this base pairing prohibits expression of maturase. I heard when I spoke earlier. Replicating opens up this base pairing, which allows A site's ribosomal binding site to bind the ribosome. It's the fact that we've replicated that allows expression of that open reading frame. So you get a small amount of A relative to the coat protein made, and indeed relative to the daughter or the progeny viral genomes. So it's the delay in the refolding of that secondary RNA structure that allows for A expression. Packaging and lysis. So going over the replication cycle. Via the pilus, the bacteriophage binds, gets to the cell surface, and the capsid doesn't enter, but there's well, we'll see it in the T phages, an injection and entry of the genome, but not the capsid within the host. We have expression of the viral protein, the coat replicase, and later maturase. Replication. We then have an accumulation of coat proteins that are going to self-assemble by binding to the genome. So coat protein has an affinity for the plus RNA. Plus RNAs get packaged into new capsids. The replication leads to maturase, which lyses the bacteria, leading to release of progeny viruses. double-stranded RNA phage. Let me just say it, there's nothing much to it. We got a segmented genome, more than one double-stranded RNA. Polyhedral core, and lipid envelope. The segments are large, medium, and small.
Sorry, is that part polyhedral?
Polyhedral. Large encodes polymerase complex, so it's more than one gene. Medium encodes the structural proteins. I think I've missed a slide here. Including spikes, which you'll see a representation of spike later. That's the attachment and injection protein, and then small enclosed that encodes the capsid and other proteins necessary for assembly. We need an RNA-dependent RNA polymerase. So this is not encoded by the host. And the plus strands are both the replication template and the messenger RNA. Missed a slide here, excuse me. That's it for double-stranded RNA. Single-stranded DNA, there are two types, filamentous and we're starting with icosahedral and filamentous. An example of the icosahedral is the microviridae, which to make S means small viruses. Phi-X174 is our example. That was the first genome sequenced, in large part because it's very small and very easy to work with. Codes proteins F, G, H, and J, which I want to know where the heck is I. F is a shell. G is a spike. H is a spike pilot. So then, yes, I can. J is DNA binding.
I can't see H.
So G is the spike. So we've seen this analogous proteins in eukaryotic viruses. H is what's called the spike pilot. These are roughly analogous to our TM and SU proteins in eukaryotic viruses, where we have a protein assembled on top of another, both of which are responsible for genome. J is a DNA binding protein, so we have the capsid protein, or known as shell. The problem here is that the nomenclature is not the same. Shell protein is the capsid protein. G and H proteins our ligand proteins that are gonna interact with host. J is a core protein that binds the genome, which is single-stranded DNA.
I'm so sorry.
You know, I'm trouble with my wife. Circular 11 point something, I think, 4 kb genome. Those are 11 proteins. 11 proteins, 5.4 kb, sorry. Encodes 11 proteins. Transcription is all in the same direction. I don't care if you remember any of this. There's still, what I'm saying I don't care that you know is, I don't care that you know that gene C is a DNA maturation protein. If you get interested in YX174, more power to you. We do have the structural proteins F, G, H, and J is out of order. So we have, that's it. I'm just going to move on to filamentous. This is the only thing to say there is this is done by rolling circle replication, which we'll see again. So we need to move on. So there's no need to dawdle. So M13 is our example of a filamentous phage. It affects through pilus. It's ingested, so it does not dump its genome, but rather enters or causes pilus retraction and then enters through the cytoplasm, as we'll see in a bit. They do not lyse cells. They will just continuously extrude new virus. They may kill the cell because they take too much biosynthetic energy from the cell, but these never cause lysis. It's a circular molecule, 6.4 kilobases is a plus strand of DNA. It also encodes 11 proteins. I didn't want to dawdle here and say, again, I don't care that you remember that protein 4 is a membrane protein for assembly. I suppose you should know they have membrane proteins. We'll talk about that in a bit. But in looking at maps, I don't know what your I don't know what your degree of facility is here, but we have P as promoter, so we have several sites, but not one for each gene. So we have what amount to polycistronic messages, so at every promoter we're going to make an RNA, and they share transcription terminator sequences. So you get different RNA molecules that have gene for protein 8 made. So for example, we're going to have a promoter here, and a promoter here, all of which may make mRNAs containing 8. 8 is going to tend to be a protein that's not needed as much. because we'll have the ribosomal entry sites coming in and translating the resulting RNA. So the virus achieves regulation of the amount of each protein it needs by having different messenger RNAs encoding different reading frames, and then distance from ribosomal entry site also affects the amount of protein made. For example, protein 3 is the one of the major co-protein or major proteins of the filament. It has its own promoter. It's going to be at the 5' end of the messenger RNA. Two, 5, and 10 involve DNA synthesis. 3, 6, 7, 8, and 9 are capsid. Structural proteins. 1, 4, and 11 are involved in assembly. I'm not going to ask you this on the final. It's going to be proteins 3 and 6 that are involved in pilus binding. Proteins 7 and 9 are going to bind our DNA, which has a packaging signal, so there's some sequence-specific protein binding there. And then protein 8 is the major filament protein. So replication of the genome occurs via host cell RNA polymerase, priming, synthesis of the minus strand of DNA making a circular double-stranded molecule that is nicked by topoisomerase and turned into a replicative form. I looked for this, but I couldn't find any information on why it does this. Topoisomerase is an enzyme, if my two fingers are the strands of a DNA molecule. It nicks one, winds it around the other, and ligates, so that you can do supercoiling by topoisomerase activity. So we have a replicative form that is supercoiled, but when replication continues in the second step, we have a nick in one of the two strands, which would tend to relieve the supercoiling, and then rolling circle replication, such that we're using the minus strand as template for the progeny DNA that is hydrolyzed by protein 2 and protein 10 into, so this is concatameric, into single molecules that we can amplify around again, or as that displaced strand with accumulation of protein 5 via translation stops the priming and synthesis of the minus strand, and toward the latter stages of infection we get the plus strand coated by protein 5 and ready for packaging. Protein 5 binding exposes the packaging signal on the DNA. I suppose there is. I don't know what it is, and based on a 3-minute literature search, nobody knows. I assume somebody knows, and it was published perhaps in the 70s, and it's just not popping up, but it's in none of the general review articles. I looked at maybe 5 general review articles on M13 trying to figure out why they have it. Super close. But your textbook is blank on the answer, and the textbook probably looked at some of the papers I looked at, nobody seems to know. Maybe there's an additional LCA assignment there. I'll have to see if there's an answer first before I think about looking for it. So, overview of the replication cycle. We have the filamentous M13 binding the pilus. Pilus is retracted to the point where we have delivery of the DNA via, this is the outer membrane, so there's a lipopolysaccharide, there's some enzyme activity here. We see that we're leaving a viral protein within the periplasm, that's protein 8. Single-strand DNA replication is the way we, rolling circle replication. In addition to replication, you're getting translation, particularly protein 5, which is the single-stranded binding protein that when it accumulates enough, keeps the progeny DNA from going back through the replication cycle. That exposition of the packaging signal allows packaging into a new virus. The protein 8 is expressed and transported out of the cytoplasm into the periplasm, and as the emergent virus comes out, protein assembles into a new virion. Then lastly, thankfully, double-stranded virus. So there are T-even and T-odd phages. I believe there are 7 T phages in total. The differences for us are superfluous. They're morphological. So they more or less follow the same patterns. Your book discusses T4 more, so will I. They are icosahedral in nature, elongated relative to a equilateral icosahedron. They have a tail structure that consists of quite a lot of different proteins, base plate fibers, sheath, and neck proteins as they're known. They're very large relative to other viruses, 170 kb, and a linear genome. Encode about 300 genes. Neither time nor inclination to go into them all. But they are examples of phage defense against viral enzymes. So among other things, some of these genes encode nucleic acid metabolism proteins, so they modify their cytidines. They modify their nucleic acids to make them resistant to host restriction enzymes. They encode their own ligases and endonuclease activities as well, and a number of structural genes. I'm looking at a whole. There's a nice animation. I had the link. It's just for a quick break.
E. coli bacterium by recognizing cell surface receptors of the host with its long
This isn't the
tail
one, but it's
fibers.
close enough.
The recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers that bind irreversibly to the E. coli cell surface. The base plate changes conformation and the tail sheath contracts, causing GP5 at the end of the tail tube to puncture the outer membrane of the cell. The lysosome domain of GP5 is activated and degrades the periplasmic
A lot of
peptidoglycan.
throbbing going on.
The remaining part of the membrane is degraded, and DNA from the head of the phage can travel through the tail tube and enter the E. coli. After the life cycle is complete, the host cell bursts open and injects the newly built viruses into the environment, destroying the host cell. Fibers.
I did want to point out the example of so one, there are a lot of proteins here, so there are a lot of genes, and it's part of the 300, but we have a number of binding, maybe there's my final example, affinities and conformational changes that occur due to binding events. So none of the phage infection of the host is active. There's no ATP hydrolysis, it's all spontaneous, and it's due to affinities in the protein. So you see that the initial contact is made via fibers. There's the base plate, I believe the base. I can't remember what they're called, we'll see it offhand. But there's contact that causes conformational change, that causes additional contact. So we have a series of binding events that lead to a tight association, and that conformational change that extrudes spike protein from the sheath. And it's this spike that's going to allow penetration of not only the outer membrane. There's a enzyme, I've lost the animation, but there's a lysozyme that will hydrolyze the peptidoglycan to allow injection of the DNA, which is packaged in here, but as the spike happens, the sheath will collapse as the spike goes in, allowing diffusion of the viral DNA into the E. coli. And then, T4 ligase is the enzyme that we use in the lab for all sorts of molecular biology. One of the odd-numbered phages. T7 polymerase has been largely replaced now, but was the original DNA Sanger dideoxy DNA sequencing enzyme. It doesn't have a proofreading capability, so you can incorporate chain-terminating nucleotides as we do when we're sequencing DNA. Another example of double-stranded DNA is lambda phage. I don't really want to say anything about this other than it's a linear genome. It can integrate into the host chromosome. But this has also been a great molecular biology tool because much of the genome is replaceable. So you can have phage that make infectious progeny that are lacking in a lot of these genes, which means we can put DNA that we want into them. Again, this technology is it's not dated so much as less useful than it used to be. We can make libraries. So I'll just end on the story. I've ever told you about hunting a cat. So when I was a eager beaver first-year graduate student, I should have known to quit the day I started graduate school because I walked into my new advisor's office. They said, I want you to get a cat and take its liver and make a cDNA library from it. Which is, we were in a blood coagulation lab and we were studying a protein called the vitamin K-dependent gamma-glutamyl carboxylase, which makes it so post-translational modification so that your blood can clot. There had never been a patient discovered anywhere with a blood coagulation disorder that had a defect in this enzyme. But somebody in the Netherlands had a cat, a house cat, that had a bleeding defect that looked like it might be in this vitamin K-dependent carboxylase. So he said, I want you to get a cat, get its liver, harvest the RNA, reverse transcribe it using reverse transcriptase, into cDNA, so clonal DNA, so eukaryotic messenger RNA, but rendered as DNA, so you don't have the introns. And I said, okay, well, where do I get the cat? He said, I got a bow and arrow you can borrow. That's when you quit. But eventually I calmed down and realized I needed to call the pathology department, and of course we're sacrificing cats for other reasons. So I went over to the cat sacrifice lab with my thing of liquid nitrogen. They sacrificed the cat, threw a liver in liquid nitrogen. I made a cDNA library. So I took the mRNA from the liver, I reverse transcribed it, and then randomly inserted that cDNA. So every messenger RNA in that cat's transcriptome, what it was making in its liver, was put into a virus. I had a pool of millions and millions of viruses, which I then infected on this is my version of a Petri dish, and they form plaques. So a lawn of bacteria, the plaques form, that comes from, if you do it right, it comes from one virus. I then used, we didn't know the cat sequence at the time. So this was, I don't even know if they have a cat genome now, but I guess they do. So this was the 90s. We did know the sequence of the cow. And so I had oligonucleotides that were degenerate, so they were my guesses as to what the cat sequence might be. And then I would transfer the viruses to a filter where the DNA would stick and probe with radioactive cow oligonucleotides. And when one would, and then I would expose them to film. So you have a marker, and if that one lit up, that means that virus had DNA I was interested in, and I could purify it back. So these libraries are very effective ways of screening entire genomes for particular sequences. And if you're working on an organism today, like Eric Albrecht in the biology department studies snakes, nobody cares about, at least nobody has yet cared enough about the sequence of genome. He's very interested in some of the genes in their venom, and he has to go look for other methods like this. So the bacteriophage are useful molecular biology tools. In the case of lambdaphage, I've personally used it to clone wild type, whatever that is, feline domesticus. There's another big question. If I kill a house cat in Chapel Hill, North Carolina, is it going to have the same sequence as the Dutch? So that other house cat was in the Netherlands. And it turned out anyway, 2.5 years went by. I made a library, cloned the gene, had flown in on KLM a biopsy from the house cat in the Netherlands. The owner would not let it be sacrificed. They actually did surgery for me. I made a library of that when I cloned them, I sequenced them, and guess what? They were identical. So after 2.5 years, I took this to my advisor. He goes, yeah, I thought that might be the case. Maybe the defect's in, there's another gene in the cycle called the reductase. Maybe it's in the reductase. It was at that moment that I quit. My mistake was going back. Well, of course, now I have no responsibility. So it turned out, well, you know, that's it. Phage will be on the final exam. Don't worry about it now. Worry about 10 through 19. I'll be around all day tomorrow.